We're going to get started today. Our first event of the day is an incredible keynote and fireside chat, and I am excited to be able to introduce you to the two folks who will be taking us through that. Dr. Victor Velculescu, world-renowned scientist, entrepreneur, founder of Personal Genome Diagnostics and Delfi. Victor's going to take us through his journey as a scientist and entrepreneur. And following his sharing his story is going to sit down with Matt Hellauer, who's the managing director at PTX Capital, to have a fireside chat and really talk about what it takes to support entrepreneurship at this scale here in Baltimore and beyond. I also want to share, because I won't have a chance to do it before Matt joins the stage, we're really fortunate to have Matt Hellauer as part of this community. He is a Baltimore native who, after spending a decade at Allen & Company, decided to come home and build something here to help grow this ecosystem. And so he founded PTX Capital, and while he is an incredible investor, performing in the top quartile, both funds, more importantly, he's an incredible human being who volunteers of his time and expertise and network to entrepreneurs across Hopkins, across Baltimore, anywhere. And so we're really fortunate to have you be part of this ecosystem and grateful for all that you do. Look forward to the conversation that Victor and Matt are going to have. Victor, thank you for being here. Please come teach us.
Oh, thank you very much. Thank you, Myra. Thanks, Myra, for the introduction. And I want to thank not only Myra, but JHTV, Matt Hellauer, Matt Tremblay, Mike Henkin, and Brown Advisory for organizing this event and for inviting me to participate today. It's really a pleasure and an honor to tell you a little bit about the story that started with our science and led to some innovation and then ultimately companies that are translating this for patients. So thank you all very much. Let me just start off by just telling you that the story I'm going to tell you today is going to start with some science, and then it's going to give you some practical examples of how we ultimately made this into tests for patients. And I kind of view this as a turning point. It's a very exciting time where the intersection of AI, machine learning, and our understanding of cancer genomics is going to really completely change, I believe, the way medicine is practiced in the future. And these are just a few examples of how we've been involved in this, I think, next generation of cancer diagnostics. So my disclosures. And then to say that all of this work began because of the groundbreaking efforts at Hopkins over the last few decades with my colleagues, with members of our team, and with many members of the community, as well as other universities. And you can see some examples of this here. These were the first cancer genomes that had ever been sequenced, really decoding and understanding the underlying blueprint of human cancer in a number of different cancer types. This led to new technologies. It led to new targets for therapeutic intervention, some of which are FDA approved, such as against PI3 kinase and IDH genes. And it led to an understanding of the underlying landscape of human cancer, the complexity of it, the fact that you need precision medicine in order to get patients the right therapies. And it also led us to realize that in order to make an impact in cancer, we need also better diagnostic approaches and ultimately screening approaches using liquid biopsies. And I'll touch about some of these things in the moments to come. So the first example of how some of this technology began to make it to patients was through founding of Personal Genome Diagnostics, or PGDX. This was founded back in 2010 with Luis Diaz, my close colleague here at Hopkins. It was based on a number of groundbreaking publications. You can see him led to a number of patents. And over that time, and I must say it did take longer, this was the first company we had founded, so it took us a little bit longer to learn things that worked and didn't work. We did do a number of firsts. Probably could have done them faster, but here they are. We had the first CLIA laboratory to do a cancer exome, first to show that ctDNA could be a universal biomarker of cancer, Luis made a beautiful connection between immunotherapy response and certain molecular changes that happen in cancer. And then we began moving into machine learning. And this was actually the first example of using machine learning to identify changes in cancer patients that didn't involve human intervention. And then this finally led to the first FDA-cleared cancer genome profiling kit. It was actually the first one that had ever hit the market. And it is sort of a landmark, I think, in the space. Now, PGDX went on to be ultimately acquired by LabCorp, a very large diagnostic company, as many of you know. And it is, I'm proud to say, still here in Baltimore. So PGDX is just down the street. It's in the Bottle Building. It used to be in Tindeco, and still has its expertise here in the area and sort of serves as one of the hubs of an ecosystem that I hope will be growing in the years to come. So this was sort of our first foray into this, but this effort was really focused on cancer patients, making cancer patients get the right therapy at the right time based on the understanding of what's happening in their tumors, but it didn't address many other aspects of how we can impact cancer morbidity and mortality. And I think one of the realizations that's happened is that despite all the efforts that we're making against cancer with new therapies, with various interventions, cancer rates continue to rise. You can see the graphs here. By 2035, there'll be over about 25 million new cases of cancer. This is worldwide, and about 15 million deaths. And so we need to think about other ways to intervene here. And it's clear that the largest potential impact against cancer will be in early detection. But for screening to work, we have to find cancers early, because it's at the earliest stages where we will make an impact on cancer mortality if we can intervene then. And so we have to get it at early stage. And then the other aspect about screening is it has to be a public health effort. It has to be available to everybody, widely accessible, inexpensive tests that are easy to utilize. It can't just be for a small fraction of the population. So a number of folks have started using liquid biopsies to begin to understand and detect cancer early. A liquid biopsy is essentially looking at DNA in the blood as it comes out of the cancer, and a number of technologies have been developed in order to try to understand what's happening there. And a number of them have happened here at Hopkins, and even within our group. You can see some examples here. I think the key is that the initial focus was on mutations. Mutations, remember, are these changes that are happening in the DNA code that make cancers different from normal cells, and that's where therapies have targeted. And that's where folks initially began to look at liquid biopsies, but we began to realize that this is just the tip of the iceberg. We're missing a lot of the changes that are happening inside cancers by just looking at these mutations. You can see the little itty bitty circle is the mutations, lots of other things that are going on. We're not getting them. So we needed a different way of doing it that wouldn't miss the DNA in this way. And then we also needed a way that was less expensive. These tests were initially made for late-stage cancer patients. In those settings, the therapies are expensive. Folks can tolerate expensive diagnostics. We needed an inexpensive test that could be applied to asymptomatic individuals. So we began to think of completely different ways to do this, and one of the ways is sort of in this analogy here. Rather than thinking of a pebble that hits the water, and that's the mutation, you've got to be there exactly the right time, you've got to see the pebble. We're not looking for the pebble, we're actually looking at these ripples that are happening downstream of that event. You can see it here on the picture, and you don't have to be exactly there at the right time. You can come later, you could see, of course, these ripples continue, and you can look at it from many different parts of the pond, which increases your chances of detection. And this was through the observation that the DNA that makes it into the blood not only has these little typographical errors, these mutations, but it actually has this broad changes in the fragmentation, or the way the DNA is cut up into little pieces. Normally it's small, the DNA that gets into the blood from normal cells in cancer, but if you look across the genome, there's actually a pattern. The pattern is very consistent in healthy individuals. That's the top line here. But the bottom line shows what happens in patients with cancer. It's just completely disorganized. And it's disorganized because the cancer cells themselves are disorganized, and this fragmentation comes from the way the cells die and they release their DNA in the bloodstream, and cancer cells, as they grow, do release and turn over. So this initial observation was made in 2017. It was kind of like an aha moment. It happened by a small team of folks, a graduate student from biostatistics in the School of Public Health, my close colleague, Rob Scharf, who's both in the Oncology Center and the School of Public Health. And I think, and I'll mention this maybe later today, that this is one of those cases where it could have only happened at a place like Hopkins. It required the expertise of lots of different individuals with different sort of areas of focus, mathematical, biological, clinical, to bring this story that I'm going to tell you together. And Rob, for example, has been a very key partner in this. Stephen led a lot of this initial work, and the other members of our group from different backgrounds made this possible. So it's a special place here that really allowed this story to happen. So we realized that with all this information, you can actually analyze it in ways that had never been done before. You can use machine learning. Machine learning takes advantage, and artificial intelligence takes advantage of huge amounts of information. Well, we had the whole genome now. We weren't just looking at a few mutations. We were looking at the whole genome and its fragmentation. These started happening in 2017, 2018. We did more things. I soon realized this is a completely new way of detecting cancer. We could actually make this into a product to help patients. So we started a company that we called Lighthouse Medicine at the time. It wasn't very creative. It was right on the water, right next to Lighthouse down in Cannes. So that was its temporary name. We then coined the term Delfi, which stands for DNA Evaluation of Fragments for Early Interception. And then that led to the name of Delfi Diagnostics. The paper that Stephen was working on and that we just mentioned was ultimately published in Nature in 2019. And it was about the same time that we began our seed round, of fundraising for the seed round. And we ultimately got a seed round in 2019. In August of 2019, Menlo Ventures led that. And you can see that around that time, we started offices here at Ashland and Fast Forward, just down the street. And by the way, fantastic now to have that facility at Fast Forward to be able to do this. It was just very difficult when we started this at PGDX. We can talk more about that. And then we also found people in California. There's a lot of expertise, of course, across the US. We aim to find the best people wherever they were. Subsequently, this was led by Series A, led by OrbiMed. Glad Rishi is here from OrbiMed today. Thank you, Rishi, for coming, and thank you for your support. 100 million Series A in 2021. A number of subsequent events, breakthrough designation by the FDA. We ended up having a lab in Palo Alto as well. We then published the first example of this in lung cancer, and I'll talk more about that. We then started a prospective trial in lung cancer. We then had a Series B, which was led by DFJ Growth for $225 million, and a number of investors, which I'll mention in a second. We ended up getting our lab certified as a CLIA-certified laboratory, and then we began showing that this technology could be used in other cancers, liver cancer in November 2022. And now let me sort of fast forward between that 2022 timeframe to today, and tell you a little bit about the focus of Delfi today and its first product that is now already out there making a difference for cancer patients. Now the goal of Delfi and its mission is cancer diagnostic tests that are high performance at a disruptive cost, utilizing machine learning, and of course are going to make an impact for cancer patients. I'm pleased to say that after a number of years of shepherding Delfi through its early stages, I was able to step back as the CEO, and I was very excited to bring in, and the board was very excited to bring in Susan Tusi as the CEO. Susan was the former chief commercial officer at Illumina and has a very stellar and distinguished background there, and we have a number of other folks that are very successful that you can see here on the C-suite. Tom Russo, Peter Bach, who actually led some of the early screening guidelines for lung cancer at Memorial Sloan Kettering before we recruited him to Delfi. Rob Geigley, who was the chief commercial officer at Invitae. And Amelia Singh, who was actually the chief scientific officer at Grail before she came over to Delfi, excited by the potential of this genome-wide approach and the impact that it has. Now, I must say, in starting these companies, one of the lessons I've learned is it's not just the team you have, but it's also the investors. And the board and the folks that are supporting you are actually incredibly important in so many ways that you don't even appreciate as a young entrepreneur. So I'm really pleased and proud of the investors that we have, not only for their support, but frankly for all the help and advice and guidance that they've provided over the years. Very strategic folks. You can see them at the bottom. They range from smaller investors to large investors, local here, Baltimore investors to investors that exist on the West and East Coast. So we're very proud of that, and I think it's actually helped a lot in a company like this. So just in a few minutes, I wanted to tell you a bit more about the science of fragmentation and then also the first product that this has led to. So the reason this approach is powerful is because it generates information that is vast. It's, I would say, a next generation liquid biopsy test. It provides more information than I think any other liquid biopsy test that's out there. I'm a little biased, but that's the sort of observation. You're getting millions of data points of these fragments as opposed to just thousands or hundreds for mutations and methylation. And when you get this information, which is of course obtained from whole genome sequencing, and I should say it's low coverage whole genome sequencing, so it's an inexpensive, easy to do laboratory process, you get lots of additional molecular information. You can see that at the bottom. And you know, this began with that Nature paper in 2019 in the top corner there, but you can see since then there have been a number of publications on lung, on liver, ovarian cancer, brain cancer, anyway, more insights in the biology, many of which are listed here. And this is based on really a lot of history that's come out of Hopkins and with the other members on the team from their experiences as well. The beauty of machine learning is that you can use more data to constantly make your approach better. And you know, a lot of discussions occur around machine learning and artificial intelligence. I think this is a beautiful example where it actually is being used in that way. So you get a lot of information from this fragmentation. The more patients we do, the more data we have to train and ultimately improve the classifier. Adding additional features, regions of the genome, adding additional analytes can help. And the advantage of this ultimately for a test is that it's an inexpensive test that's always the same, the laboratory procedure. The difference in the different applications have to do with the machine learning algorithms and the training that occurs. So there's a great potential for continuously improving it. We focused as a company on lung cancer screening, and I'll tell you about that in a minute, but there is early and I'd say proof of concept data in liver cancer, multi-cancer, other single cancers like ovarian cancer, brain cancer, and others that are underway, monitoring patients during the cancer care continuum and even in minimal residual disease. And the reason we focused on lung cancer is the greatest cancer killer worldwide. It remains so today despite great efforts in smoking cessation. You can see here some statistics about it. Very deadly disease, more cancers, more deaths from lung cancer than many other cancers combined. And the sad thing about this is we know that early detection can be helpful, but we are not doing it today. Low-dose CT can be used for this, but it's only about 6%, maybe 10% these days, but it's extremely low nationwide, and especially in areas like Baltimore. Unlike many other cancers where screening rates are high. Now, we chose to use this technology in this setting, initially in a proof of concept experiment led by somebody here at Hopkins, Dimitris Mathios, in our group, and you can see the results on the left. The very bottom row are patients with cancer, very disorganized fragmentation profile. The top two are patients who have either no cancer or benign lesions. And it's really important to be able to distinguish actually benign lesions. A lot of biomarkers don't do that. So very clean, consistent profile across the chromosomal arms, and this is the fragmentation profile, while very disorganized here. Now I'm pleased to say that that concept, the proof of concept, has now, and that was done in collaboration with colleagues at another institution in Europe, and a very homogeneous population. So the idea was, well, that's nice, but let's do it prospectively. Let's evaluate this test directly. Let's do it in a population that's in the United States, diverse and across many centers. And pleased to say that this work has now been validated in a prospective clinical validation, over 1,000 individuals, case control study, 48 different centers in the United States, led by Peter Mazzone, one of the world experts in lung cancer screening, and Peter Bach. And you can see the same exact patterns that you saw in this proof of concept. And that has now led to a test which Delfi has created for exactly this purpose. It's called First Look Lung. It is available through a number of different health systems already. And the goal of this test is to evaluate the intended use population, individuals above 50 with a history of smoking, They're not getting screened today. Could a simple blood test be helpful here? And this is where it fits in. If you're positive, you go down the typical diagnostic pathway. If you're negative, you go to annual screening. The key with a test like this is to have a very high negative predictive value. You don't want to turn somebody away from this proven and health-saving, potentially, diagnostic path if you're not really sure that they're negative. So this has a negative predictive value of 99.8%. It's higher than the 99.5, which is recommended for this cancer. And it is now already being used, as I said, by a number of health systems. And the simulations or sort of projections are that even a modest uptake nationwide would decrease cancer deaths by thousands of individuals over a 5-year period. So I'm very excited that this story, which I began for you today, started off with science, built here at Hopkins under, I'd say, this unique ecosystem that we have, led to the birth of this company. And now in a period of about 5 years, a test which is now clinically available. There's still a lot of work to do. This test is on its way in multiple clinical trials to get FDA approval, to be evaluated in Europe. These trials are actually have completed, and it's in the sort of follow-up stage and analysis period. And ultimately, I think it'll be wonderful to see this become something that's standard of care, and then move on to other applications. So just a brief summary. We talked about fragmentomics and the ability to then turn this into accessible screening tests. We now have shown that you can validate this particular technology, and we have a test that's actually useful for lung cancer. And there is the great possibility of applying this, I think, in this population of individuals that are at need. And I think the excitement here is that there's a platform. The platform is the same, and you can apply it really across the cancer care continuum and in many other cancer types. So anyway, that was my brief introduction. Sorry for being a little long-winded, and I'm happy to now take any questions and chat more. Mara, can I get my water? There's a water down there.
Thanks so much, Victor. It's an honor to share the stage with you today and have you in Baltimore and to be a partner of yours. We're going to spend about 30 minutes having a conversation, the two of us, and then we'll open it up to audience Q&A for the last 10 minutes. And in those 30 minutes with you, Victor, I want to unpack 3 things with you. One is you as a human, clinically trained scientist and an entrepreneur, a luminary in your field, and I think an equally excellent, impressive manager and CEO of your companies, which is a rare combination. The second is the design decisions you made at Delphi in those early days to make this such a formidable company in a competitive market. And then the third is the liquid biopsy market today and why it's so relevant to so many of the therapeutics community in this room. So let's start with you, Victor. Let's rewind the clock a bit. You grew up in Romania in the 1970s. It was a complex time in Romania. It was caught between the East and West in the Cold War. Tell us about your childhood and any formative moments that got you on the path you are today.
Yeah, no, sure. So I was born in Romania, and I came to the U.S. when I was very young. I was 7 when I landed actually in California. But I do have some very sort of striking memories from Romania. One of them was that around that time, some of you may remember, there was a major earthquake. It was like a 7-point-something earthquake in Romania. A lot of devastation. A lot of folks needed help. We had family members and friends that were physicians and in the clinical field. So it always instilled in me a desire to want to help and to help those who need it. And I think the other part of it that I remained with even when I was in the U.S. was the desire to do, to make an impact clinically that could help anybody that needs it. And maybe I'm taking this too quickly to this place, but it was one of the things that drew me to Hopkins. You may know, and some of you may remember, that Hopkins Hospital actually was founded as a hospital to be available to everybody, regardless of their backgrounds, regardless of socioeconomic levels, race, and so forth. It was something that was incredible at the time. So I've always felt this desire to make things that could be useful by everybody in society, especially those that need it most, and even those in other parts of the world that could benefit and whose lives could be improved.
You came to Hopkins in 1992. for your MD, PhD. You haven't left since.
Some say I never graduated. It's fine. I'm really enjoying this place, and I'll keep staying here.
Victor's twins are undergrads at Hopkins, so they might graduate before you.
That's right.
And tell us about that path at Hopkins in the early days. Why not pursue the clinical path of the MD, and you went down the discovery path in research to form companies? Why?
Yeah. So, I mean, Hopkins, first of all, has, as many of you know, an incredible graduate, undergraduate, and medical school, School of Public Health, anyway, a number of different schools. I won't belabor it, but in particular, the medical school, which I'm familiar with, has a program which is an MD PhD program. You'll do an MD and a PhD. It's sort of simultaneous, and you get a good exposure to clinical medicine, but also science. And I had the fortune of having some fantastic mentors during my time during the PhD. The experience was very impactful for me, and I thought that I could make a greater impact by essentially focusing on the science rather than the clinical work. So I continued. I did complete my MD PhD, but I did not then pursue clinical medicine. I continued in research, and then that's led ultimately to the realization that in research you can only get so far. You write papers, the papers are nice, but how do you get them to patients? And you can get them by licensing, of course, to larger companies. Sometimes that just takes a long time. And you're one of thousands of things that they're considering to license. The other is to start your own companies, which will then make a technology ultimately approved regulatorily in a way that can make it to patients. So that was sort of the path that I've taken, and this has been a great place to do it.
And what got that entrepreneurial bug in you from 92 on? What did the entrepreneurial ecosystem look like at Hopkins back in the 90s versus today?
I mean, I think, as I mentioned at the very beginning, I think there has been a sea change during this last, I'd say, several decades here at Hopkins. I think JHTV, with Fast Forward, the place where you can incubate companies, the expertise that folks now have at JHTV has been greatly improved since what it was, I think, in the 90s. And, Hopkins, I would say historically, was extremely well known for its science and for its grants. It's like the best funded university from perspective of NIH grants among many in the world, but it was not well known for translating these to companies, especially in the area. And so it was challenging. It was challenging. I think initially there weren't many examples of this, but I think we persevered, we succeeded, we learned through a lot of mistakes. It was great doing this with a partner like Luis Diaz. And after that experience, of course, it's easier. It's always easier the second time. And it's also become much easier because I think this realization that we need to improve this. And I think credit goes to President Daniels, Christy Weichsel, many others that have put a lot of time and effort to make this process better, more informed, easier. And then, of course, you need investors, you need experts that can join companies like this as they're starting, and I think that investment ecosystem has improved as well. Obviously with folks like you, with Brown Advisory, with many others that are local. So anyway, it's made a big difference, I think, since when we started this.
That's excellent.
Yeah.
And let's talk more about that discovery process in your lab. Your technologies are so commercially relevant and attract interest from those beyond the scientific community. PGDX raised over $200 million and then sold for over $500 million to LabCorp. Delfi has raised over $400 million, is tackling the #1 cancer killer in the United States. When you are in the lab, are you working backwards from an end clinical market, or are you day-to-day in the science and then figure out that market eventually?
Yeah, it's a great question. One of the benefits of doing this, of doing research in a place like Hopkins, and in particular in my lifetime where I was exposed to a lot of the medical part of Hopkins through the medical school, you realize that you want to do science, but I wanted to do science that would make an impact for patients. And if you ultimately do something that makes an impact for patients, then you'll do something that could lead to a clinically useful test that'll be commercially viable and interesting and attractive as well. I think many times folks get caught up in developing a technology but not really thinking about what the application of this is. I think there's many places where this is more common than at Hopkins. I think Hopkins, we do have this unique perspective of what is clinically useful. What's really the problem you're trying to solve? If you're just doing a technology for the sake of it, it may not end up ever being useful. So I think that's the combination here that's, that's important. I mean, we're obviously interested in the underlying science and basic biology, because that can lead to new discoveries that are important therapeutically or even diagnostically. But I think having this perspective is in general been very valuable for me.
In 2 words you said there, you said useful and accessible. I think there's some concern in the medical community with liquid biopsy tests that they might be neither. There might be not a great clinical reflex when you get the results back, and some of these tests are overpriced and not accessible to the people who actually need them. Grail's Gallery test is priced at $950 out of pocket. Exact Sciences is in the $700 range. Where does Delphi fit into that, and what design decisions did you make that could compete in this market with these question marks?
That's a great question. So I think, the issues, and by the way, Grail deserves a lot of credit for being the first company out there sort of commercially pushing liquid biopsies in a way that hadn't been done before. They did a great job with that. They educated the market and so forth. Exact has been, of course, out there with a stool test for colon cancer. They have done tremendous work in educating colon cancer screening and deserve credit for that too. However, I will say that as example, because you brought it up, Grail and the Gallery test did is they took an existing old technology and they said, let's retrofit it for screening and early detection. It was bisulfite sequencing of cell-free DNA. That test currently does not have the performance metrics to really be a screening test for any of the common cancers. As an example, in lung cancer, where we know quite a bit about this, the sensitivity of that test is about 10%. In the latest real-world trial, they detected 1 out of 11 lung cancers. So that is inadequate. You would never send somebody to get a test like that if your goal is to detect lung cancer, because you have an alternative that is so much more sensitive, the low-dose CT. So unfortunately, that current technology does not have the performance characteristics to actually be a recommended screening test really currently for any cancer. And then on top of it, it's expensive, and you want tests for screening to be something that is a public health effort. You want it to be something that this population of 15 million who needs it for lung cancer, many more for colon or ovarian and others, are receiving. And you can't do that at a $1,000 price point. Just simple math will tell you that'll break the healthcare budget that we have. There are some savings from screening, but they're way, they're overshadowed by the cost of the test. So I think when you're thinking about these public health problems, you have to then start and say, well, what technology can I develop that's inexpensive and different from what we've done before? And that was the approach we took. We said from the beginning, look, none of this technology that exists today is ever going to be useful for screening. It's just not good enough. It's too expensive. Let's do something different. And that led to this this sort of breakthrough in the fragmentome that I was talking about earlier. And the benefit of that is that it's an inexpensive test. It's low coverage, whole genome, very simple laboratory work. Rather than $1,000, it's in the few $100 range. And that's something that over time, I think, it's sustainable in the US and likely worldwide.
And are you actively having conversations with payers and the regulatory bodies and telling that differentiated story and that clinical reflex that works versus others?
Yes, we of course are. And the team is, of course, now doing that directly. And I think the great thing is it's hugely positive response. I think when people see that you're trying to do something that's actually going to make an impact in public health, everybody wants you to win. The regulatory, we've had incredibly positive interactions with the FDA. They want this to succeed. They see this as a need, and this is probably the only thing that's, and I didn't mention it before, but this is the first liquid biopsy for lung cancer that's actually ever been approved for clinical use, I think, worldwide. So they see this as something that could be the first FDA-approved test for clinical screening of lung cancer. And similarly with, I think, health systems. Health systems want to help patients. They can't afford to do the most expensive things all the time that are not that helpful. And so when they see something like this, high-performing, low-cost, it actually makes sense to start to implement. And so some of them, even without FDA approval, which leads to reimbursement, or USPSTF approval, which we're seeking, we're starting to get folks that are actually, paying for the test, health systems that are subscribing to it, and so forth, which is a wonderful thing to see so early on. It's almost unheard of, honestly, in screening and diagnostics.
Mm-hmm. And let's talk about that path to winning. It is a very competitive market out there. Every big player that Delphi is competing against or compared against is having 10,000 plus prospective clinical trials out there. Grail has divested itself from Illumina and has had a quiet but great year in the public markets, doubling its stock price since January. Guardant is a $7 billion company now. There's a lot of money here, even if the story does make the most sense to regulatory bodies. How does Delphi win in such a competitive market?
I think it comes down to fundamentals. I mean, you want a test that's high performing and inexpensive. I think, having completed these large trials that we have, again, the readout has to come. But if they continue to have the same performance that we've had in the case control studies, the same performance we've had earlier on, I think you'll end up seeing folks gravitate to a test that's better and less expensive. So, and I should also say, nobody's currently, Delphi, from my perspective, appears way ahead in the lung cancer space. Mm-hmm. Nobody's sort of doing this in lung cancer. Grail is multi-cancer, not useful for the common cancer types, perhaps useful for rare cancers. We can talk more about that, but and then of course Exact is focused on its multi-cancer test, but also stool, colon cancer detection, and Guardant is currently interested in colon cancer as well.
Yep.
So Delphi's sort of alone in this space. I would say that there are folks that are interested in lung cancer as well, of course, and some of these companies have mentioned that. But I think it has an advantage by being the first that's out there, much farther along in terms of the trials that it's done,
yep.
And so far with fantastic performance.
And let's talk about that multi-cancer versus single cancer world now. It is a bit confusing to the market that both of these worlds are going on parallel paths. Can they coexist when the dust settles on the industry?
Yeah, so it's a little confusing. There are obviously detecting cancer early and intervening is thought to help save lives for individuals who might have cancer. Now, it's been demonstrated in a handful of cancer types. So lung cancer, colon cancer, breast cancer, and cervical cancers are the ones that the U.S. Preventive Services Task Force has given a grade of an A or a B, saying that these early detection and intervention in these cancers will save lives. For other cancers, we believe that screening and early detection will be helpful, but we need more evidence. So, there's some evidence, for example, in liver cancer, quite a lot of evidence there, or gastric cancer. A lot of societies recommend that too. But for many others, not there yet. So I think some of these companies initially, Grail as an example, said we're going to try to do everything at once. We're going to try to do all these cancers at one time. And the challenge with that is that the cancers that early detection helps, and where screening tools already exist, have to be considered in their own sort of world, because they already have existing screening tests. You have to do at least as well as they are, perhaps better, and you have to make a test that makes sense from a clinical perspective. So our approach to this has been, let's take the right clinical question, let's focus on that, yep. Perhaps one cancer at a time for these cancers where screening makes a difference. And that's the approach we've taken for lung cancer, and we will likely take for other single cancers. And over time, we'll also be having a multi-cancer test, perhaps separately for these cancers where you are still exploring the benefit of this. And in those settings, those cancers are rare. They need different performance characteristics. They need to be highly specific. These other ones, you need to be highly sensitive, because you don't want to miss something that would be caught by an existing test. So I think you need different approaches. I think we have a two-tiered or multi-tiered approach of thinking about that. And I think that also has the advantages of fitting within the current clinical guidelines, the regulatory guidelines, and sort of getting to acceptance of these tests more broadly. So I think it has many advantages. Mm-hmm. Yeah.
And talk to us more about that traveling story for the fragmentomics technology. Liver cancers next. What do the tests beyond liver look like? And then can you see fragmentomics expanding beyond cancer to other diseases too?
Yeah, great question. So liver cancer, let me just pause for a minute and talk about liver cancer. I didn't show any slides on this, but very excited about the impact of this potentially in liver cancer. Liver cancer is the third leading cause of cancer death worldwide. It is a major problem in areas where individuals have cirrhosis or hepatitis. There's about 400 million people worldwide that have these conditions and are at risk. A lot of them in Asia, but it is actually rising in Europe and in the US, and it's because of the foods we eat. It's tied to what's called fatty liver disease, and these new syndromes of Maslow and others are risk factors ultimately for fibrosis, which is then a risk factor for cancer. The reason that's important is it's on the rise. We need a way to detect it and intervene early. We've had an initial study that was published in 2024 showing great performance for detecting liver cancer, led by Zach Foda here at Hopkins. The performance of that looks to be now repeatable in additional cohorts that he's done from around the world. And so it looks like no matter where you have liver cancer, you end up having the same fragmentation profile. So again, it's the same exact test, a different classifier based on different machine learning, let's say, regions that the machine learning takes advantage of. So anyway, we're excited about getting that across the finish line at some point in the future. We think there's a great need for it. There are other cancer types where we've shown this can be successful. We've had examples in ovarian cancer. Ovarian cancer is one of the deadliest cancers, of course, in women, and there's no great biomarker. It's just a terrible disease. In fact, there have been trials, very large trials done with existing biomarkers like CA125, and they've all failed. And part of the thinking is that hasn't had a high enough performance, a positive predictive value. So we've shown that the combination of this approach with existing biomarkers, now with CA125 and another protein, can actually have higher positive predictive value. So ovarian cancer, I'm personally very excited about, and we're doing a lot of work on, still I think in the research phase. Brain cancer has been a cancer that nobody thought could be detectable as liquid biopsies. You've got the blood-brain barrier. It essentially blocks things going back and forth, and essentially it's been a failure in the liquid biopsy space until, from my opinion, this most recent study that Dimitris Mathios, a former neurosurgeon here at Hopkins, led using the technology, and he showed essentially unprecedented sensitivity for brain cancer detection. So anyway, lots of opportunities in multiple cancer types. And as we've talked about before, I think there is an opportunity even beyond cancer. We presented at a recent AACR meeting that the technology could be useful in detecting changes in the liver. We were talking about liver cancer. Before you get liver cancer, you get what's called liver fibrosis. Detecting liver fibrosis is a major thing now because there are drugs for liver fibrosis and potentially cirrhosis in the future. So we've shown that you can detect that using this, which is a precursor, of course, to liver cancer. So I think there's opportunities in that realm of liver disease, but also other conditions which lead to changes in the blood, which, again, with machine learning can lead to tests that will be useful for patients.
Very good. I'll do one final question, and then we'll open it up to audience Q&A. You're very much a diagnostics guy. There are a lot of therapeutics investors in this room, and you've also attracted therapeutics investors into your cap table. You have at Merck, BMS, Eli Lilly, between PGDX and Delphi. Why is Delphi and going upstream to early detection so important to the therapeutics community?
That's a great question, and it's very interesting. So, as many of you know, a lot of the therapies that exist today, whether they're targeted therapies, chemotherapy, or immunotherapy, have some effect, of course. Immunotherapy in particular, everybody's very excited about, but most of the time, actually, they still fail. And they fail not only at the very beginning, when you give them to patients, but they fail because patients get resistance. They'll respond initially, but then the cancer comes back. So why does that happen? It happens because the cancers at later stages have time to continue mutating and changing and having ways to escape these therapies. So the beauty of targeting these cancer patients early is that they don't have time to develop all these things. And this is something that a lot of researchers have identified, researchers here at Hopkins and at other institutions. Luis Diaz, my former partner, had a trial recently where he showed that if you use immunotherapy early on in colon cancer patients with mismatch repair deficiency, 100% of the patients respond. It's a much more impressive response than if you wait for late stage. So this has gotten everybody realizing who's in the therapy world that, well, if we detect cancer early, that'd be helpful, which, of course is something we've been thinking all along. But now we have multiple avenues of reasons for that. Not only could we intervene surgically, we can intervene from a therapeutic perspective. And so many companies that have invested in Delphi, Merck, Eli Lilly, and others are very excited about this aspect of the technology. Helping identify patients earlier means better responses to their therapies.
Excellent. We'll open it up for the audience. We'll go to David Rothschild in the back.
Wait, hold on.
I'm going to ask an economic question, which is the holy grail for companies like yours is getting to reimbursement.
Yes.
But that's not the holy grail. The holy grail is incentivizing utilization. And clearly you get better clinical outcomes, hopefully, by detecting early. And my question is, for the carriers to incentivize their cohort of providers to actually use your diagnostics, they have to be excited about the economic consequences. So I'm curious if earlier treatment and better clinical outcomes leads to less intensive medical intervention, lower cost treatment, et cetera, et cetera. And is that kind of how you're framing it? Because that's how I would want to frame it, which is, I don't care if you're reimbursed for $9 million if you only get 15,000 people taking the test.
I think that's a great question, and obviously utilization is very important on sort of multiple levels for this and many other aspects. So a couple of things just as background. One is you have to do, in many of these settings, in order to convince us all that these kinds of tests are useful, a clinical utility study, and Delphi is actually already doing that. So we have large-scale prospective screening trials in the US and in Europe, they're completed, but also a clinical utility study that's underway. And that shows that you're doing the test, and the test then leads folks to go downstream and do the next step to get low-dose CT and others and to pursue the path. So that's one layer of evidence. But the bigger question is, how do you persuade clinicians, health systems, et cetera? I think the fascinating thing here, and it was a lesson for me as well, is when you have a test that's that's inexpensive, let's say, not nearly as expensive as others, and you model out where the money goes when you are talking to a health system, you'll actually see that there's a return on investment. And the reason is because the positive test leads to further downstream work within the health system, all of which, of course, is reimbursed. Of course, you're saving lives. You're saving the patient's life. You detect them early. But the health system keeps more money inside their health system as opposed to patients coming in very late, where most of the money actually goes to pharma because they go to the drugs, right? So if you detect it early, there can be surgical interventions, there's biopsies, all sorts of other things you need to do. So essentially with a price point that's this low, as Delphi has and it's accessible, there's a positive ROI. And that's been the argument with a number of these companies and a number of these health systems, and they've begun doing it and paying for it directly, which is, as I mentioned earlier, actually, it's essentially unheard of in the diagnostics world. Most of the tests come in too expensive, and everybody waits until reimbursement. Yeah.
Next question.
That's a great question.
Ben Scruggs.
Thank you. Great way to start the day. Thanks for sharing. The aspects you mentioned of starting with PGDX and lessons learned, are there any things that you can extrapolate on that relative to how you've moved things quickly with Delphi? And also thinking about LDT pathways as you interact with regulators of additional tests beyond lung and where those lessons are taking you and building the company going forward.
Yeah, and I think part of it you just touched on. I think, for a diagnostics company to ultimately become profitable, you need to get reimbursed. And so moving on that pathway as quickly as possible is important. It took us a while at PGDX to figure out what that pathway was, and so that took some time. I think part of it is with expertise. And I will say that the environment in Baltimore is growing, and I think has changed a lot since when we started PGDX. So there's a lot more expertise in all the areas that you need in a company. In the case of Delphi, we said from the very beginning that we want to just find the best people everywhere. It happened actually during COVID that we were fundraising, and so we ended up getting a site in California as well. There's expertise that was scientific here, there was expertise that was regulatory, reimbursement, and so forth there and in other parts of the country, so we just found the best people wherever they were. So I think sort of getting experts that are knowledgeable in converting your science into a product and then converting that product into a regulatorily approved test, whether it's CLIA or FDA approved or internationally approved, is really essential. And those were some of the lessons learned for me. Yeah.
We have time for one or two more questions.
Victor, first of all, thanks for your decades of leadership in this area. My question is really about that you mentioned the application. So you chose negative predictive value, but actually, if you thought about positive predictive value, in some ways, if you wanted to get people into this funnel and get them a low-dose CT, I'm just curious why you chose NPV to get them out of the funnel versus PPV. I mean, certainly GRAIL has this challenge that PPV is only about 40% from the Pathfinder, but in their case, you send patients down a tremendous diagnostic odyssey because it's a multi-cancer detection test. But in your case, even a false positive, basically you're taking this high-risk population into low-dose CT, which is not a bad thing because the compliance is low single digits. So I was just curious your team's thoughts about NPV versus PPV.
I think you said exactly right. I think the goal in lung cancer screening is to get as many people in, to not miss individuals who might have lung cancer. And if you have a false positive in this case, the next step is low-dose CT, which frankly they should have been getting anyway. There's no harm done. And then you sort of continue down the path. In order for that to work, you need to have a really high sensitivity. And the other way of saying that is you need to have a really high NPV. When you tell somebody they're negative, you have to be really sure they're negative. You don't want false negatives. So it's really the same thing. It's a high NPV, which reflects the high sensitivity of the test. And the sensitivity, for example, the Delfi test, about 80% across all stages, largely stage 1, which is very high, while the GRAIL one was 10% or so. So that's like the 10% is unacceptable. You can't have that as a screening test because you're essentially telling people not to go on for normal screening because you think they don't have that condition, but they very well could because you're missing the vast majority of them. So an NPV corresponds to exactly what you said. You need a high PPV if you want to prevent a diagnostic odyssey. So let me give you an example of that. And that's why I was saying earlier, you want a different kind of clinical scenario to guide your test and to have a different test for different conditions. In ovarian cancer, there's no great imaging test that happens next. You actually, in many cases, need to do open laparotomy in order to find what is there. And so the guidelines suggest that you need a PPV of at least 10% in order to proceed to that next step. This is a really rare cancer. You don't want to send lots of women to be getting laparotomy when it's obviously totally unnecessary. So in that particular setting, you need a very high PPV. And the reason that that's acceptable is there's no existing diagnostic or screening test for ovarian cancer anyway. So any amount of, you could have a lower sensitivity, but as long as your PPV is very high, that's still valuable. Anybody that you detect with ovarian cancer is acceptable, if that makes sense. So there's sort of different scenarios, and it's very important actually to keep these in mind. It took us a long time to figure out what is the target product profile. What is it you actually need for this particular condition versus a different one? And it's crucial to having these companies ultimately make the right tests.
One final question. Yes.
Yeah.
Yuka.
Yuka Manabe from the Department of Medicine. Great talk. And just to say, Peter Bach was an Osler Medical resident to continue
Yes,
the Hopkins
nice.
Odyssey. So I want to continue on that vein. I love the concept of a companion diagnostic where you make a diagnosis, you screen, you figure out that the person has a risk, and you go on to do another diagnostic test. But what do you do with people if you have a very sensitive test where you might find something before the next screening tool, the next tool that you use, you leave them in limbo. They're worried, but their next test doesn't come back positive. Have you done sort of talking to patients about what they would do with that information because they feel like a ticking time bomb?
Yeah, I think that's a great question. And Yeah, first of all, Peter Bach has had an extraordinary influence on, I think, this whole story, and his medical training was here, and he's just been a great thinker of this whole field. So it's been wonderful to have him. I think there's going to be many tests. These tests, when you think of screening, are not the same as the test that ultimately gives you the final diagnosis. Screening tests are meant to guide you into a pathway. And so we do this all the time. We have high cholesterol, we do all sorts of tests that mammograms are a good example. The first test may be positive, but the subsequent one ultimately is negative. And that's accepted just because you're used to the fact that many of these initial ones are false positives, but there's no harm done because you're essentially just getting a follow-up. So I think it's partly patient education and educating ourselves where in this pathway these tests fit, and getting used to the idea that right now nobody's getting screened. 95% of folks are not getting screened for lung cancer. Getting them into this pathway and increasing the numbers that are getting screened, having the test work in the vast majority of cases. Of course, there are going to be cases where there may be nothing to do after it's positive, but that's just going to be part of the way a population scale test works. And we just have to educate ourselves about that.
Thank you for the questions. I'll end with one final one, Victor. There are many budding entrepreneurs in this room and also co-investors that are building alongside those entrepreneurs. What is one piece of advice you'd like to leave everyone with of taking a technology and commercializing it into something that can have impact positively on a global scale?
Well, that's a tough question to end on here, but first of all, I think they're all fortunate because they're in an ecosystem where they can make this happen, as we've just talked about. I think the unique environment here lends itself to doing things that are going to be life-changing and let's say clinically changing for the world. But as you build your company, I think probably the most important piece of advice I could give you is focus on the clinical question and get the right team around you. And the right team being not only the folks you hire and bring in, but also the investors that work with you. I think bring a set of folks that are not just bringing resources, but are helping you along on this journey.
That's great. I came home with the hope of meeting people like you, and it's an honor to be a backer of yours and most importantly, a friend. Victor, thank you so much for the time.
Thank you very much, Matt. Thank you.
Okay, I'm about to release you to network and grab a beverage. A couple of